We continue looking at cell division here, and what today is about is taking a look at what happens to the DNA throughout the cell cycle, because the DNA changes. DNA is synthesized, as we learned in the cell cycle diagrams last time, during the S phase, and then and that's in preparation for the cell undergoing cell division. So the DNA is probably the most important thing that daughter cells get from parent cells through the process of cell division. It's genetic information, it's hereditary information that's passed from parent cell to daughter cell, and also from parents to offspring. So that's why we're studying cell division and heredity together in this unit, because cell division is how hereditary information is passed on. So these diagrams are all showing in photographs, because these are micrographs. This is a transmission electron, or I'm sorry, yeah, this is a transmission electron micrograph. This is a scanning electron micrograph of chromosomes. So they're all representing chromosomes, but we're going to start with this diagram. And first thing we're going to do is relate this diagram to the cell cycle. So this part A of the diagram here, where it says one chromosome unduplicated, that represents the form that DNA is in during interphase. And remember that interphase is divided up into three phases. So specifically G1 is when we find the DNA in this form. So every molecule of DNA, notice that this is labeling one molecule of DNA. So this is one molecule, this is one molecule, this is one molecule of DNA. But in G1 of interphase, for each molecule of DNA, there is only one copy of it. All right, so that's what we refer to as unduplicated. So in the G1 phase of interphase, each chromosome is unduplicated, one DNA molecule. We also refer to that as single-stranded. And the other thing here is that not only is it unduplicated, just one molecule, but it's also uncondensed chromatin. And hopefully you remember back when we were studying cell structure and function, what we labeled on the inside of the nucleus of the cell was chromatin. And I made that point then that a cell that is not undergoing cell division has DNA in the form of chromatin, not chromosomes. Okay, so in interphase, in G1 of interphase, the DNA is unduplicated. Each DNA molecule, there's only one copy of it, and it is uncondensed chromatin. Then, of course, we go through the S phase, which is synthesis of DNA replication, and that's where we go from one copy to two copies. So during S phase of interphase, before a cell divides, each DNA molecule replicates. And so we're only looking at one DNA molecule here, but when that DNA molecule replicates during the S phase, then we have two molecules of DNA. And so this represents the DNA in a cell in G2 of interphase. So we're still in interphase, G2. But notice that there are two DNA molecules, and we refer to that as double-stranded. So these are two DNA strands together, and they stay together. They're attached together. And each DNA molecule is referred to as a sister chromatid. A sister chromatid making up what is going to become a condensed chromosome. But at this point, it's still uncondensed chromatin. In G2, it's still uncondensed chromatin, but it is now duplicated. All right, so we go from unduplicated single-stranded DNA molecules to duplicated double-stranded DNA molecules. Now there's a copy of all the DNA in the cell, and that is in preparation for cell division. Following G2 then is cell division, and that begins with mitosis. Remember, mitosis is nuclear division, and the first phase of mitosis is prophase. And that's when we see the uncondensed chromatin condensing into chromosomes, what we actually can see under the microscope as chromosomes. And that's what this scanning electron micrograph is representing this transmission electron micrograph and this diagram. The chromatin condenses into what we call a mitotic chromosome, a condensed mitotic chromosome. And we're going to look on the next slide what this condensation means. What do we mean by condensed? How do we go from uncondensed to condensed? We'll look at that on the next slide. But the other thing to point out here is that these two DNA molecules that make up one chromosome are attached to each other at the centromere. So that's the point of attachment between two sister chromatids. So the two DNA molecules are referred to as sister chromatids. So notice that for every chromosome, there are two sister chromatids, and we're about to divide, and they're in a parent cell that's about to divide into two daughter cells. So hopefully you can see where we're going with this. Each daughter cell is going to get one copy of DNA. Each daughter cell is going to receive one sister chromatid from the parent cell, from each chromosome from the parent cell. So when a cell divides, the sister chromatids separate so that each daughter cell gets one copy of each unduplicated chromosome, which uncondenses back into chromatin. In other words, we go back to G1. So now all the DNA that's in that cell, the daughter cells, are going to be single-stranded, unduplicated, uncondensed chromatin. And the last thing is something you'll want to remember. They're only referred to as sister chromatids when they are together making up one chromosome. Once they split, as soon as they split during anaphase of mitosis, you no longer can call them sister chromatids. You have to refer to them as unduplicated chromosomes. So when they're making up a duplicated chromosome, they are sister chromatids. But once they split, they themselves then are simply unduplicated chromosomes, one DNA molecule. So what is this condensing all about, this condensation? Well, this diagram pretty much shows how the DNA is packed into chromosomes. So we're starting here with a DNA double helix. So that's a molecule of DNA, which is only 2 nanometers wide. That's 2 billionths of a meter. That's not very wide at all, but DNA is a polymer of nucleotides, if you'll recall, polynucleotide, and the molecule can be very long. And if you consider all the DNA in one human nucleus, all the DNA in one human nucleus, if you put it end to end, would stretch 6 feet. So there's a total of 6 feet of DNA in every human nucleus. So it's not a very wide molecule, but it's a very long molecule. And so because it's so long, you can imagine it would get all tangled up, like trying to hang Christmas lights when they're all tangled up into a bunch. So the cell has to organize the DNA somehow. And that's what these things are doing. So histones are proteins that organize the DNA. Each histone makes up a bead on a string. We call this beads on a string, right? And it kind of looks like that, especially in the diagram. But it looks like that under the electron microscope too. Each bead is 8 histones, 8 histone proteins. Even though you can't see them individually here, this bead represents each 8 histone molecules that are bound together, and then the DNA wraps around that. And that entity, once the DNA wraps around those 8 histones, is referred to as a nucleosome. All right, so that's a nucleosome, and it's made up of 8 histones, 8 proteins called histones, plus the DNA that's wrapped around it. That's a nucleosome. And that's the basic packing unit of DNA. The next thing you need to understand about this diagram is, and I would recommend you put a dotted line on your copy, above the dotted line is considered chromatin, right? So DNA is in the form of chromatin during interphase. In other words, G1, S, and G2, all the way through interphase, the DNA is in the form of chromatin. So you'll notice in the diagram, this is labeled chromatin, and this is labeled chromatin. Once we cross this line, what we're looking at is a chromosome and then zooming in on how the chromosome is constructed. This is what we're talking about when we talk about condensation. When the chromosome condenses, the chromatin folds up, coils up into a chromosome. That's condensation. So during prophase, and that happens during prophase of mitosis, the chromatin condenses into mitotic chromosomes. That's what we're looking at here, is a mitotic chromosome that's visible under the light microscope. So before prophase of mitosis, you can't see chromosomes in a nucleus, but as soon as the cell gets into mitosis and begins nuclear division, that's when the chromatin condenses into chromosomes, and you can see the actual individual chromosomes under the microscope. So chromosome, condensed, mitotic, right? So this is a mitotic chromosome that has been condensed from chromatin. So chromatin condenses into chromosomes by wrapping and packing and folding and coiling. And even so, it would be easy for the DNA to get all tangled up. And it's amazing and kind of mysterious as to how the cell keeps the DNA organized and keeps it from tangling up. So as I pointed out on the previous slide, what we end up with here is a mitotic chromosome that is duplicated, made up of 2 DNA molecules. So that's how you know this is a duplicated chromosome. Each DNA molecule is a sister chromatid, and it's attached at the centromere. So these are sister chromatids. And remember, when the cell's undergoing mitosis, nuclear division, these sister chromatids are going to separate. Once they separate, then we refer to them as unduplicated chromosomes. So just to finish up here, there's the centromere, and what we have at the end is a duplicated mitotic chromosome. Now let's look at the cell cycle and actually look at, there are micrographs here. These are light micrographs of a real cell undergoing cell division, actually undergoing the cell cycle. But then there are diagrams along with each one, so you can get a better idea of what's going on inside the cell. So this is the cell cycle. It's not just mitosis, it's the cell cycle. And so we start here in interphase. So this all represents interphase in between cell divisions. And we've got cells here where we have unduplicated chromatin. So it's not only unduplicated, but it's uncondensed. And so that would be G1. So these cells are in G1. And then we go through, of course, S and G2. But notice that I also labeled the centrosome with the centrioles here. And notice the difference as we go from this cell to this cell. There's only one centrosome with centrioles, and here we have 2 centrosomes with centrioles, and that's because we've gone through G2 and the centrosome has duplicated. Remember, everything duplicates after the DNA replicates in the S phase, then everything else doubles and duplicates or replicates during G2, and that's why you can tell a cell that is in G2 because it has 2 centrosomes. Not only is that cell in G2, but because it went through S and G2, that means it's a cell that's about to divide. Therefore, we would refer to it as a parent cell. And because it has passed through the S phase before the G2 phase, that means that the DNA contained within that parent cell is duplicated chromatin. It's still uncondensed as chromatin, but the DNA has been duplicated. So there are 2 copies of every DNA molecule in that parent cell. And that would be considered then the parent nucleus. And then we get into, mitosis, which is the beginning of cell division. And notice that this cell is in prophase, the first phase of mitosis, and the nuclear envelope's breaking down. We can see these duplicated condensed chromosomes, so these are mitotic chromosomes. We also see that the centrosomes are moving away from each other. The 2 centrosomes are moving away from each other and forming a structure in between them that we refer to as the spindle. So we've got duplicated mitotic chromosomes now. That's what happens during prophase. They condense so that they are visible under the light microscope, and this is the way it looks in a real cell. Then we get into metaphase and notice that the chromosomes, the nuclear envelope has completely broken down. The centrosomes have reached opposite poles of the cell and completed setting up the spindle in between them. And the spindle fibers that make up the spindle are attached to the chromosome, have attached to chromosomes and move them to the middle of the cell. And again, meta means middle. So the chromosomes are lined up along the middle of the cell in metaphase. And then we have anaphase, and ana means away, and you'll notice that the sister chromatids, each of these duplicated chromosomes is made up of sister chromatids. Here in anaphase, the sister chromatids have separated, and so now we no longer can refer to those as sister chromatids. They are now unduplicated chromosomes, and they are moving away from each other. There are two groups of them, or sets of them, and they are moving away from each other, one from each chromosome. One unduplicated chromosome from each duplicated chromosome that we started out with is now moving to opposite poles of the cell. And then we get into telophase, and in telophase, the nuclear envelopes reform, and we end up with daughter nuclei, and that's the end of mitosis. So this represents the end of mitosis, which is followed by, of course, cytokinesis, the actual splitting of the cell. And again, cyto means refers to cell, kinesis refers to motion, movement, and so it's the actual movement of the daughter cells as they split. So now we have the daughter cells, which then enter back into G1, and the cycle starts anew for each one of those daughter cells. On the next few slides, we're going to take a closer look at what happens in each phase of mitosis. So here we have prophase, and we're going to start off labeling everything. So, of course, this is a parent cell that's undergoing mitosis and cytokinesis, cell division overall. So we've got the cytoplasm, the plasma membrane, and it's a good exercise to label these whenever we have a diagram like this to remind yourself of what everything is and the terminologies, because I throw around terminology rather quickly as I'm describing things and explaining things. And of course, we've got the parent nucleus and the mitotic chromosomes, the condensed duplicated mitotic chromosomes, made up of sister chromatids. So each chromosome has two sister chromatids attached at the centromere. And that's just simply the point of attachment between the chromatids. We've got the nuclear envelope surrounding the nucleus, but it's breaking down. So being represented here as a dotted line because it's breaking down. This is a centrosome, and there are centrioles within the centrosome, a pair of centrioles, if you'll recall. And the centrosome, if you'll recall, is a microtubule organizing center. Back when we were studying cell structure and function, that's the function that we assign to the centrosome, that it's a microtubule organizing center. And that's what it's doing here during cell division. It's organizing microtubules. These red lines here represent microtubules that are going to form the structure known as the spindle. And the spindle is made up of two parts, basically. There are the asters, which are these short microtubules here that are actually anchoring the centrosome to the plasma membrane. So that's their function. They anchor the centrosome to the plasma membrane once the centrosomes have reached opposite poles of the cell. So these centrosomes haven't quite reached opposite poles of the cell yet, but so that's the function of the asters that are part of the spindle. And then we've got the spindle fibers, the spindle microtubules. So you'll see them referred to as fibers also. So either spindle fibers or spindle microtubules. I think it's better to refer to them as spindle microtubules to remind you of what they're made out of. They're made of microtubules. So are the asters. You could call these aster microtubules also. So here's the explanation of what's going on during prophase. Duplicated chromatin condenses into duplicated mitotic chromosomes. Made up of two sister chromatids attached at the centromere, the nuclear envelope begins to break down while the centrosomes, each with a pair of centrioles, begin to move away from each other, forming the spindle between them. And we'll pick it up next time looking at the rest of mitosis. In other words, metaphase, anaphase, and telophase.